Introduce Dr. Elizabeth Matsui, who's going to speak to us today from Austin, Texas. And I'm sorry that Elizabeth wasn't able to come and be with us in person. She's a great colleague and a real expert on this topic, but gratefully agreed to present remotely. So please have a look at the screens, and Elizabeth, take it away.
Well, thank you so much. I greatly appreciate the invitation, and I'm so sorry I couldn't travel to Vancouver. It sounds like it's been a fantastic start to the meeting so far, and that I'm missing out also on some really great food. Maybe in the future I'll be in Vancouver. So a little bit of background about me so that you have a sense of the perspective that I'm coming from, which is that I did a pediatric residency, so I'm a pediatrician, and then I did an allergy immunology fellowship. So I'm a pediatric allergist immunologist, and I continue to see patients, mostly kids with asthma. And then during another bulk of my time, I lead a research program that has been focused on the intersection of environmental health, in particular airborne exposures and respiratory disease disparities. And most of my work until the past 5 years has been focused on indoor exposures, and then more recently, some of the collaborations I've had have extended the work to outdoor air pollution exposures and respiratory health. So that's the lens that I bring to this. I am not an exposure assessment scientist, but I work closely with them. So with that introduction, let me share my screen, and I want to make sure, I'm assuming I'm going to start the slideshow, and someone will let me know if there are issues seeing the screen.
We can see it.
Okay, terrific. So before I start, I do want to mention that this is a massive topic, and that there could be days devoted just to this topic. And given, that this is really a focused talk, I'm going to touch on a variety of points at a very high level, and try to wrap up with some sort of principles about how we think about the scientific evidence related to health impacts of indoor air pollution, and some of the gaps in evidence that need to be filled to have a real impact. And so there will be things that I think will clearly not be touched on. So I think one compelling question that is particularly germane for this group, and my hope is it becomes more front and center for the public at large, is indoor air the new water? And there's been a lot of talk about regulating indoor air, and this provides sort of the lens through which we think about the health impacts of indoor air and indoor air pollution, because that often helps drive the discussion around policy change. And related to that question is trying to understand whether a variety of indoor environments are considered healthy or not. And that's a very complicated question, as this audience well knows, and not necessarily so straightforward. And part of the reason it's not so straightforward is this one very simplified schematic already illustrates how complicated the indoor environment is, and the variety of sources and opportunities for outdoor air to come indoors and also contribute to indoor air pollution. And then clearly there are a whole variety of different factors that contribute to indoor air pollution. We have indoor and outdoor originating pollution, microbes that include microbial components, as well as infectious and non-infectious microbes, allergens. I'm an allergist, so you will hear about allergens. And this is not all-encompassing or exhaustive by any means, but I hope it gives you a sense of the complexity of indoor environments and indoor air pollution. Much of what I'm going to talk about is drawn from Chapter 6 of this recent National Academies of Sciences, Engineering, and Medicine report on indoor exposure to fine particulate matter and practical mitigation solutions. And so a lot of what I'm going to talk about is focused on PM. I do sort of mention gases, but I want to kind of acknowledge that there's a whole discussion that can be had there about their health effects that I will probably get short shrift in my presentation. So importantly, all of our biologic systems are affected by exposure to air pollution. And I'm going to draw a lot on the outdoor air pollution literature as needed because for a variety of reasons related to how much more accessible outdoor air pollution data is than indoor air pollution data. A lot of what we understand about potential indoor air pollution health effects comes from the outdoor air pollution literature. And we know that outdoor air pollution contributes to indoor air pollution. And so it's really not that much of a leap to frame our thinking about health impacts of indoor air pollution through the lens, at least in part, of outdoor air pollution. And so the biologic systems include the lungs, the heart, the endocrine system, the neurologic system, as well as reproductive system. And you can see in bold are actual sort of diseases or health conditions that have been linked to air pollution exposure. And then the ones that are not necessarily in bold are health-related outcomes, but are not necessarily sort of a disease or a diagnosed condition. And so this alone also makes it difficult to, or more challenging to study the health effects of air pollution exposure. There's a huge amount of evidence pointing to the biologic plausibility of air pollution health effects. I have an epidemiology degree, so I also think in terms of, what is the sort of burden of evidence that's needed to demonstrate causality, particularly when at least for outdoor air pollution, we don't have randomized clinical trials available. So when we're thinking about observational data, and there's very strong evidence that it's highly biologically plausible that indoor air pollution causes adverse health effects. So the mechanisms that have been elucidated or proposed include oxidative stress and inflammation, direct cell damage through toxicity. Certain sources of PM, like combustion, will elicit a specific type of inflammatory response. In this case, neutrophils, so a type of white blood cell that you often see when there's a bacterial infection, but that is a hallmark of PM exposure. And air pollution may also exert effects beyond the respiratory tract through penetration to the circulation, and this is more applicable to the smaller size fraction. Particles have been found in the placenta as well as in the central nervous system. And so obviously particle size has a lot of influence in terms of the site of impact and the mechanism of impact. And then the mechanisms that are well described for certain bioaerosols are that allergens, if they act via an allergic mechanism, are acting through the presence of an allergic antibody that someone has to that allergen, specifically IgE that's specific to the allergen. And then certain microbial components like endotoxin act through the TLR4 receptor and other molecules along that signaling pathway. So across all different types of sources of air pollution, there's clear, strong evidence of both biologically plausible mechanisms and biologic effects that would absolutely be expected to translate into clinically relevant effects. And I mentioned this earlier, and I think this was a really important point, and it's something that came out of the National Academy's committee work, was that some of the evidence that was discussed in Chapter 6 and in other parts of that report relied on outdoor air pollution. And it makes a lot of sense when we're lacking indoor air pollution evidence to look to that body of evidence, because human beings spend the majority of their time indoors, and outdoor air pollution or pollution of outdoor air contributes to a meaningful degree to indoor air pollution. So, a word about sources and composition. One thing many, many years ago when I was doing my fellowship training that was really a very basic concept, but was fascinating to me was, there were some environmental engineers talking about PM, PM, PM. And I said, well, what is it? And when they describe that it's simply sort of particles and the composition of it was not known, from someone who comes at these issues from a biologic and clinical perspective, that was really surprising to me. And of course, over the past couple of decades, there has been, growing evidence that the source of PM and its composition may matter, as it may confer different types of toxicities. And I know that the theme of this workshop is really about wildfires. And I want to mention that there's several papers published suggesting that wildfire as a source of PM, may confer greater toxicity, particularly for respiratory effects. And then also there's a paper related to eczema flares or skin effects, so that the source probably does matter. And it's also important to consider the biologic sources and components of PM. So oftentimes I think how people who do research in this field have emerged has really been into they're not camps so much, but there's a group that really thinks about PM and thinks about particle count or mass concentration of PM and maybe metal or inorganic composition. And then there's kind of another group of folks who think about the microbial components or the allergens and the bioaerosol components. And we really need to think about them together because they both confer health effects and they likely interact. And I'll show you a little bit of evidence suggesting that that's the case. And then, of course, I've focused a lot on particles. Gases, as I mentioned, are also important. NO2, indoor NO2 from gas appliances. Maybe it was about a year ago. There was a paper that came out on this topic that led to many, many headlines and lots of discussions about gas stoves and respiratory disease. So we think about the health effects in terms of the acute health effects of pollution and the chronic health effects. And for the acute health effects, we think about when someone has respiratory disease, so maybe they have a diagnosis of asthma, they have known asthma, when their exposure to air pollution increases, are they at greater risk of developing symptoms, for example, or an exacerbation? So that's an acute effect. Or even in a population that doesn't have respiratory disease, do acute changes from day to day in air pollution exposure have acute sort of day to day, same sort of temporal scale effects on health. And we think about them separately because we have to study them differently and they have different implications. And so there's very strong evidence, not just in the outdoor air pollution literature, but in the indoor air pollution, that indoor air pollution causes pulmonary inflammation. So meaning infiltration of white blood cells into the lungs. I mentioned neutrophils in a previous slide, decrements in lung function, and adverse clinical effects of symptoms, asthma exacerbations, COPD exacerbations. And there's very strong evidence that indoor air pollution increases susceptibility to upper and lower respiratory tract infections. So influenza, COVID, RSV, and acute visits for these infections. And there's evidence in populations with and without lung disease. For chronic effects, these are more challenging to study because this is a question that's really framed in terms of if a child is born and over a certain period of time or within a critical developmental window has higher exposure to air pollution, are they more likely to develop asthma? And that's because, of course, you don't develop asthma out of the blue on one day. This is really an example of a chronic health effect. And we have very little evidence in the indoor air pollution world or literature about its effects on chronic respiratory disease. And this is just a practical reason why we don't, because outdoor air pollution studies can take advantage of large administrative health data sets and use modeled air pollution exposure assessments to link to those cohorts, or actually enroll large cohorts but that don't require home visits and direct measurement of indoor air, and can use modeled outdoor air pollution exposure metrics to follow them over time and examine the relationship between chronic exposure to outdoor air pollution and the development of respiratory disease. So when we think about indoor air pollution, to do those studies of thousands of children, we would have to be able to measure the indoor air pollution exposure for, a huge number of children, which is prohibitively expensive, and also just extremely logistically challenging. There are certainly new technologies that this audience will know well that might make it easier to assess the indoor environment in a way that is less labor intensive and can be scaled. But nevertheless, we are really kind of stuck right now with evidence based on outdoor air pollution studies. And for outdoor air pollution, there's strong evidence that it increases the risk of both COPD and asthma, and that it affects lung function trajectory, meaning it restricts lung function growth in children and it increases the rate of decline among adults. So in children, I'll pick one lung function measure, forced expiratory volume in 1 second. So it's the amount of air that someone can blow out in the first second that they're kind of forcefully blowing air out of their lungs. That increases over a time in children until they reach their 20s, and then it plateaus. And then in adulthood, there's a predictable rate of decline in FEV1. So the average adult will lose 25 milliliters of FEV1 each year. So you can imagine if you are a child and your lung function growth is restricted and your plateau of FEV1 is lower when you're in your 20s than it would have been otherwise, that you are starting out at a disadvantage, and your risk of developing lung disease in adulthood is greater than it otherwise would have been, because you're going to have this inevitable decline, which puts you at risk for COPD, and FEV1 in adulthood is actually a predictor of mortality as well. So there is strong evidence that one particular source of indoor air pollution with reduced lung function growth among children. So that is a chronic respiratory outcome where we have good evidence for indoor air pollution. There's a whole talk could be given about about schools, about occupational settings. These are very important to consider, particularly given the time spent in these settings. And then children are a vulnerable population, and that they are in a susceptible window during their course of development. And the example, obviously, that I just gave about lung function growth, I think illustrates that. Occupational settings are better studied and regulated than school settings. It's clear that schools can have high concentrations of air pollution, and that there are specific types of sources of exposure, like proximity to certain sources like idling buses, that produce air pollution that can come indoors as well, that are unique to schools. Schools in disadvantaged communities have high concentrations of pest allergens, endotoxin, fungi. These have all been linked to adverse respiratory outcomes, and again, acute outcomes. And then mitigating factors, so building characteristics have not been well studied, such as filtration and ventilation. And you can see here, this is a headline from a story that was in The Guardian about the location of schools of children in the UK, and those schools being located in areas where the air pollution was over the WHO guideline limit. There's susceptibility factors. So clearly, very young people and very old people are known to be more susceptible to outdoor air pollution. This figure here is a figure from a paper from our group that's now more than 10 years old, showing obesity among children being a susceptibility factor for PM 2.5 respiratory symptoms. So this was among a cohort of kids with asthma. So PM 2.5 increases along this axis, BMI increases along this axis. And then as the two increase is where the probability of having asthma-related symptoms related to PM is maximized. So there's an interactive effect here. So this is just one outcome, but we saw this with multiple outcomes. There's emerging evidence about the role of diet and omega-3 versus omega-6 fatty acids, antioxidant composition of the diet and supplements, oxidative stress gene polymorphisms have been implicated in outdoor air pollution exposure studies as being susceptibility factors, and then biologic components. So someone have allergic sensitization to an allergen that is a part of the indoor air where they're living or working or playing, and then their polymorphisms in the toll-like receptor 4, which is the receptor that interacts with endotoxin and also associated pathway molecules. So lots of susceptibility. I'm going to address health equity in a future slide because there are other aspects to vulnerability and susceptibility than just these. And I mentioned that the indoor environmental sort of mixture of contaminants is complicated when they have interactive and sometimes paradoxical effects on health. So this was a paper also a little more than 10 years ago from a cohort of kids in Baltimore with asthma. And what I show here is this is the odds ratio and 95% confidence interval of having an acute visit for asthma, having oral steroids for asthma, which is a sign of an exacerbation, and then having bronchodilator reversibility, which means that if they take albuterol, which is a rescue medication, they have an improvement in their lung function. So it suggests there's some physiologic issues going on in their lungs. And when air nicotine is below the limit of detection, the relationship between endotoxin exposure and the acute visit is actually protective. And there's a whole story about endotoxin under certain conditions and for certain outcomes being protective. But when you can detect nicotine in the home, and this is, of course, associated with us having a smoker in the home, there's really you lose the protective effect. And in fact, for these other outcomes, oral steroids and bronchodilator reversibility, the exposure to endotoxin becomes a risk when there's a smoker in the home or you can detect air nicotine. You know, this is one tiny piece of the complexity of everything that's in the indoor environment, and it's meant to just illustrate the complexity of studying the health effects, and that in some cases there are certain things that are not always harmful. And this is related to the timing of exposure and the level of exposure. So this audience may have heard about, being exposed to high levels of endotoxin in the first year of life reduced the risk of developing allergy and asthma. But being exposed to high levels in an occupational setting like a swine facility can make people really quite acutely ill. So this level of complexity makes it difficult when we think about, how best to intervene. Cardiovascular health effects. So there's most of the evidence we have is related to outdoor air pollution. And it's clear outdoor air pollution causes heart attacks, heart failure events, strokes, cardiovascular deaths. There are clear mechanisms of these adverse health effects on the cardiovascular system. I mentioned some of them here. The indoor evidence of the effects of indoor air pollution on cardiovascular disease is more limited. There's evidence from smoking bans that those have been associated with a significant reduction in cardiovascular events. You can see there. And there's more work that examines surrogate markers of cardiovascular outcomes. And that's because even though cardiovascular adverse outcomes are common across the large population, when we have cohorts, when we study indoor air pollution that are on the order of 100 or 200 people, they're not going to be statistically powered for, having a heart attack or some other acute cardiovascular event. So what I show you here is one example of a paper like this. So this was from Meredith McCormick and her group, and they followed a cohort of just under 100 adults with COPD who were former smokers and looked at the relationship between indoor PM 2.5 and a variety of surrogate markers of cardiovascular outcome. In this case, they looked at heart rate variability. So the more variable someone's heart rate is, that's a sign of better health. And right now, just ignore the non-obese and obese. These being below zero tell us that there's an estimated negative relationship between PM 2.5 and heart rate variability. So every twofold increase in PM 2.5, this is the percent reduction in heart rate variability. And notably, it's pronounced among those with obesity, and illustrating yet another sort of complicated interaction. And so this is not direct evidence of cardiovascular disease, but heart rate variability has certainly been a robust outcome for a robust marker of adverse cardiovascular events and health. There are other health effects. I'll touch on these very briefly. Lung cancer, and there are also other cancers that have been linked to outdoor PM. We know less about those cancers in indoor PM for the reasons that I mentioned. Endocrine effects, so diabetes, maternal and infant health effects, low birth weight, preterm birth, and then from the outdoor literature, the neurologic and psychiatric adverse health outcomes. So I mentioned indoor air pollution and health equity. There's no question that indoor air pollution exposure varies across populations. Communities of color and or living in poverty are more likely to be exposed to different types and higher concentrations of indoor air pollution. And that National Academy's report did not include low and middle income countries. So this is a really important area. And there's a whole body of work that I don't have expertise in, but probably there are folks in the audience that do, that are related to, biomass burning for cooking and heat in developing countries, and interventions to try to mitigate the adverse health effects in those contexts, which are particularly borne out by women and children in those countries. So these groups have disproportionate burdens also of different types of diseases associated with exposure to air pollution. So that then increases their risk of health effects of air pollution. So there's this bit of this positive feedback loop that it potentiates the adverse effects. And the effects may be amplified by inequities in context and circumstances, and you see here. Intervention. So I have just one slide. So there's incomplete data that quantifies how reduction in indoor air pollution, PM specifically in air pollution, improves health across a range of health outcomes. And the context of trying to intervene is often as important as a mitigation measure itself. So I think when you think about it from an exposure perspective and from a PM perspective, we know PM is bad, ergo reducing PM must be good. That logical conclusion often doesn't hold because the implementation of the intervention in and of itself is challenging and is an important research question. Oftentimes an intervention works in a laboratory setting and fails to reduce the contaminant that you are interested in a field setting, and then sometimes reducing the contaminant in the field setting doesn't bear out in terms of health improvements for a whole variety of reasons. Maybe the contaminant wasn't reduced enough, or it was not as an important exposure in that particular population as something else. And then there are different strategies, of course, for interventions, building and individual level interventions, and I think you're going to hear about some of these in the next slide. And so I will wrap up with this slide. So it's very clear that indoor air pollution causes adverse health effects. There's variable strength of evidence among different health outcomes. So I think the acute respiratory effects, the evidence is very strong. And then there's strong evidence of cardiovascular disease. And then much of the other connections, we rely on the outdoor air pollution data. So we have lots of engineering tools that can reduce indoor concentrations of air pollution. But as I mentioned, the interventions, when you actually go and test them in clinical trials, have variable health effects. And that's because the context matters a whole lot. There are challenges to this work, which is that in the outdoor air pollution world, there's a regulatory framework in the US and elsewhere that provides at least guideposts for what are the kinds of questions that need to be pursued and the studies that need to be done in order to align them to influence regulation. And then I mentioned that large scale studies are lacking, but which exist in the outdoor air pollution world. And we need tools to be able to scale the smaller studies that have been done and significant funding in this area. And to end on a positive note, a great example of success is smoke-free legislation in Scotland. So there was smoke-free public legislation that was implemented in 2006 that was associated with marked decreases in asthma-related hospitalization among children. This was followed by smoke-free vehicle legislation, which associated with reduced respiratory hospitalizations among kids less than 5. So it is possible to implement interventions that have really marked public health benefits. Thank you. And I think I'll leave it to the moderator about whether we have time for a couple of questions or not.
Thanks very much, Elizabeth. You probably couldn't see us, but there was a standing ovation for you here. So we'll take a couple questions. I don't wonder if there's any online questions maybe for a start. Nothing? Okay, so questions in the room.
Please use a microphone, otherwise Elizabeth will not be able to hear you.
Okay.
Hi, Elizabeth. I'm glad we could work out your presenting even remotely. I was going to ask this to Bill Nazaroth, but I think it fits for both of you, and I'm stealing a little bit from Sarah, or she can correct me if I'm wrong, but this idea that pollution levels indoor, let's just use this 50%. So let's say they're 50% lower, and most of the regulations, of course, relate to the outdoor air, and we're doing these studies, but people are mostly inside, and so we're saying, well, at a level of, say, 10, there are X health effects, but in reality, that's really telling you what the health effects at a level of 5 is for most people, because they're mostly indoors, right? So I guess what I'm asking is, if you buy that basic framework that we're making regulations and we're framing things around outdoor air pollution levels, while people that are being studied in those studies are actually indoors at about half that level. What does that say about, is that just like, okay, fine, it's 10 outdoors and we know that's 5 indoors, and so it's just a scaling factor and who cares what the real number is? Or is it a problem that we're making claims about, oh, the safe level is X, when it's really half that indoors? Do you see what I'm saying?
Yes. So, yeah, and I appreciate this question because I want to be clear that in talking about outdoor air pollution, I don't think that that evidence should be used to say something about a specific concentration of a specific contaminant in the indoor air and some quantity of health outcome. I think and so there's a difference between, I think, saying this really tells us because some percentage of it, and it varies, right, gets indoors depending on building characteristics and all of that. And we know that even at levels below the EPA standard or the WHO standard, that there's evidence that PM 2.5 has adverse health effects. So we really think this is a problem indoors. I think is not something that can be immediately actionable, except for to say this is a problem and there's an urgent need to understand it better. The other aspect that's missing from, drawing that line of logic that you just drew, of course, is all of the other things that come from indoor sources. And then you also mentioned the amount of time. And so when we think about a chronic exposure, we think about the amount of time that you're in a place, right? So the value may be 10 outdoors, but it may be 5 indoors. But if you're spending 10 times more time indoors, then, it's not probably fair to say it's equivalent to 50 indoors, but I think you're getting at my point. And so I think there's a nuance here, which is that, we should be highly concerned about our gaps in direct evidence regarding indoor air pollution and health effects, given what we know about outdoor air pollution and health effects, and that outdoor air pollution on average, makes up a meaningful proportion of indoor air pollution, and people spend a large amount of their time indoors. Does that help answer?
No, I think it's awesome. I only say one other thing. Part of it is about communication, where people get this idea that, the scientists have said 10 is bad. And in reality, indoors it's actually a lot lower. But those are the same people that are being studied in those studies that say 10 is bad. So anyway, I agree with what you're saying. I just think there's also this communication thing where people misunderstand what safe levels are based on the fact that the science is predominantly outdoor, but studying people who are actually indoors at a lower level and nonetheless having these adverse health effects. So that's a whole nother, I guess, risk communication thing. But I appreciate that, and I won't go on further.
No, thank you. That was a terrific question and comment.
Thanks. We'll take maybe one more question. I'm going to ask it then, if I don't see anything. Which is, so one of the things that's always troubled me, I guess, is, I think for logistics reasons, much of the indoor air quality health literature is sort of focused on people with pre-existing conditions. What do we know, and how do we get to the point to actually understand, for the general population, the health impacts of, let's say, indoor air quality?
That is a big question and such an important one. And I think it goes back to this kind of practical issue of, so if you have a general population, the health outcomes of interest are going to be less common. So you need larger populations. And so this goes back to the issue of scale. So we clearly have, I think about this as an equation. You have X on the exposure side, Y on the health side. We have the ability to measure health at scale. But we have less ability with the exposure side of things when we're talking about the indoor environment. And so it's not just going to be one solution. Like, even if there was a massive study that, was incredibly well funded, there's just a logistical problem with scalability. And some of the folks like you will have thoughts about the ability of, having remote monitors or consumer grade monitors, and whether that will get us to where we want to go. And are these things that, from a study perspective can be shipped on a wide scale and monitored, and we feel like the data is, good enough in order to start, oh, look, my balloons were on. I don't know if people saw that. Good enough, to do an epidemiologic study. Now, on the flip side, we do have the ability to do interventions in a way that we don't with outdoor air pollution. That's also a scalability problem, but we've been involved in a few small size clinical trials where we deploy HEPA or HEPA-like air purifiers, and if you can get, a 50% reduction in indoor PM, and again, this is not a general population, but you do see a reduction in asthma symptoms in pediatric kids living with a smoker. And so that's a more powerful tool sometimes from a study design perspective. But with the general population, you would need a large population, and it would depend on the frequency of your health outcome. But I would be curious what your thoughts are too about that, Mike.
No, I think it's a challenge. I guess it's also also a communications issue that I would say the inability to demonstrate some of these health impacts is more because of the lack of appropriate study designs, not because of the lack of evidence. So everything that we know about the science and the understanding, and I think Bill Nazaroff's talk covered this quite well. Indoor air is just as messy, and if not messier, than outdoor air. So I actually just sort of take a precautionary approach to it. But as you said, there are examples from with newer monitors and surrogates and things that I think we can get scaled studies. So, but challenging topic.
Right.
So thanks very much. I think we have to move on. Again, wish you were here to join us, but thanks so much for a wonderful presentation.